First, I need to thank you for coming to the UTB students today. And I need to appreciate the support from LEC and biology department. This seminar cannot happen without you. And we know communication is an important part in scientific training. Although EEPB, the Ecology, Evolution, and Population Biology program, is one of the sub-programs of DBBS, which includes up to 500 students. There's not much interaction among us, maybe due to different research topics or just the physical barrier. We hope to break this isolation, at least within the program. In the past, students might feel scared of giving talks at research-day seminar because we feel we need to be very well prepared to show some fascinating results, like those professors and postdocs do, which is a good ambition. However, we graduate students are often in the middle of the way. It may take too long before we can prepare well. So this time, we want to make a change. In a conference-style talk, graduate students can share their ideas, hypotheses, or preliminary results. We search for any feedbacks, questions, or suggestions. I believe they will be very helpful for developing our thesis project. Today, we have 3 courageous students to take the first shot. They are Justin, Baldwin, Sean, McHugh, and Rhiannon, Vargas. Please join me to welcome them.
Hello, everybody hear me well? Okay, thanks for the introduction, Lynn. I'm Justin Baldwin. I'm a 4th-year graduate student in Carlos Montero's and Jonathan Myers' labs. And I'm going to talk to you today about how birds have adapted to thermal gradients. This is a project that we have recently resubmitted together with Joan Garcia Porta, a former postdoc in Carlos's lab. Yes. Is there a way to do this better? So this is my outline today. I'm going to talk to you first about some ecogeographical rules. You probably all know Bachmann's and Allen's rules specifically, then get into what I think of as subtle complementarity between the 2 rules. And last, we're going to look at some of what drives families of birds to either conform to Bachmann's or to Allen's rules. So to start off, I'm going to get into what these rules are. We know that life on Earth is found in extremely hot as well as extremely cold environments. And across the planet, endotherms have adapted to this geographic gradient in temperature in lots of different ways. Carl Bachmann in 1847 was one of the first biologists to really deal with this question of how endotherms, that is animals that make their own body heat, have adapted to temperature gradients. And he identified that the ratio of an animal's surface area to its volume is a particularly important factor that governs heat exchange. And specifically, he noted that as animals get larger body sizes, they can attain low ratios of surface area to volume simply because their volume expands faster than their surface area does as they get bigger. Conversely, animals with small body sizes can attain higher surface area to volume ratios. And they should also be found in predictable parts of the planet, right? So places with cold temperatures should sport assemblages of animals that have large body sizes, presumably because the low surface area to volume ratios that they have helps them retain body heat. Conversely, warmer parts of the planet should be characterized by animals with smaller body size, presumably because the high ratio of surface area to volume that they have helps them dump excess heat when it gets too hot. And so he not only proposed this mechanism, but then went out and tested it and found lots of different lines of evidence supporting it at multiple phylogenetic scales, specifically both lots of evidence within and across species. 30 years later, Joel Allen expanded on the same fundamental logic here, but instead of focusing on body size, he focused on appendage length. So appendages, stuff like arms, legs, tails, ears. And he identified that here as appendages get longer, the surface area to volume ratio can increase because the surface area increases faster than the volume does when an appendage gets longer. And here we're not talking about we're really focused on the kinds of appendages that are particularly important for heat exchange, such as the bill of birds, such as this toucan bill, or rabbit ears. Those are highly vascularized tissues that can either transmit body heat effectively towards the outside or function as thermal windows. So conversely, animals with small appendages can attain different surface area to volume ratios, and they should all be found in predictable parts of the planet, right? Hot parts of the planet should be characterized by animals with long appendages, while colder parts of the planet one should find animals with shorter appendages. Again, presumably due to this function of surface area to volume ratios on energy budgets. So if we fast forward to the last 175 years of studies or searches for empirical support of this, there are lots of compelling empirical studies that have cemented the place of these two rules in high school biology, right? They are part of the canon of ecogeographical rules, and there's lots of compelling charismatic taxons that conform to them, right? Moose, cacti, and marine copepods all conform to Bergmann's rule. Desert and Arctic hares, as well as different populations of song sparrows, all conform to Allen's rule. However, these are large-scale rules, so there are also numerous studies that fail to find support. Right? Not every biologist who goes out and tries to find support for these rules ends up finding them. And so some of this controversy is due to methodological differences or some semantic issues arising from the translation, mistranslation, and misinterpretation of the original texts. But we think that most large-scale tests of Bergmann's and Allen's rules have really gone about this in this kind of isolated, siloed fashion, in the sense that they've gone out and tested these rules independently from one another. So, you can go to the literature and find authors that seem to go and collect one data set and then publish two papers, like these two here. We then also kind of have been looking at some of the more recent literature that documents how animals have been responding to climate change. And so these are microevolutionary, intraspecific changes of how animals have responded to temporal gradients in temperature. And there's a growing body of literature that suggests that animals have been not only changing their body sizes, but also other parts of their morphology, a phenomenon that has been interpreted as shape-shifting, right? And so this leads us to ask our principal question here, which is, have endotherms adapted to global temperature thermal gradients via perhaps subtle and complementary changes? So what do I mean with subtle complementary between Bergmann's and Allen's rules? I like to explain it like this. If animals are faced with a temperature gradient and they are somehow evolving to attain beneficial and presumably adaptive surface area to volume ratios, when they change their body size, they end up conforming to Bergmann's rule. When they change their appendage length, then they conform to Allen's rule. With subtle complementarity, we are referring to subtle, small changes in both body size and appendage length at the same time that might individually fly under the radar of statistical significance, right? These might be very weak changes individually, but if they happen simultaneously, they can both function in attaining the same end result, right? Which is the same adaptive surface area to volume ratio. So to kind of sum up our idea here, we think that if Bergmann and Allen's rule kind of represent alternative modes of adaptation, we think that an unexplored opportunity might be that animals might be adapting to both at the same time, but just weakly so. So for example, you kind of just like go out and look at birds, for example, right? We can see that owls definitely conform to Beckman's rule. We got a great gray owl in the far north, one of the largest sized owls here, and then a bunch of like lots of tropical tiny-bodied screech owls in the montane tropics. They definitely conform to Beckman's rule, but not to Allen's rule, right? Their bill size does not change. In strong contrast, we have New World flycatchers shown in blue here that occupy as, again, large temperature gradients, but they do not change their body size, but they change their bill size, right? We have stuff like boat-billed flycatcher in the lowland hot tropics of South America, and stuff like tufted tit tyrant in the cold southern cone. Ducks also occupy a similarly large portion of the temperature gradient across the planet, but barely change their body size and barely change their bill size. So we think ducks are an example of subtle complementarity here, or shown as conforming to neither rule. So you can kind of guess what we did, right? We went out and quantified conformity to both Beckman's and Allen's rules at the same times across 107 families of birds. We covered almost 70% of all bird life, about 7,000 of the world's 10,000 species. And we excluded about 3,000 species because we really wanted to focus on non-migratory resident species, right? As well as terrestrial species. We did that because we wanted to be able to accurately quantify the thermal niche that they experience, right? The average temperature that a species experiences. We then fitted two phylogenetic linear mixed models. The first one that allowed us to quantify the conformity of each family to Beckman's rule had body mass as a response variable, as a proxy for body size, mean temperature as a predictor variable, and then we allowed for phylogenetically pooled random slopes of temperature. We did the same approach to quantify conformity to Allen's rule, where we analyzed bill size as a function of mean ambient temperature, again, allowing for the same phylogenetically pooled family level random slopes. We used kind of a bunch of metadata bases to get at phylogeny, morphology, body size, and range distribution, and include global climate as well. So then, after fitting these two models, we extracted those phylogenetically pooled random slopes. And so what I'm going to show here is a plot of plots. Right? On the y-axis, we show how strong a family conforms to Allen's rule, but that is how hard do they change their bill size given a certain amount of temperature change. On the x-axis, we have the strength of Bergmann's rule, right? Which is how strong does family change their body size given the same exposure, given the same change in temperature. And so each point here is going to represent a family. And so what we see is that 10 families conform to Allen's rule, shown here in blue. About 8 families conform to Beckman's rule, shown in red. But 89 families conform to neither, shown in gray. So this suggests that there are largely, there's a restricted mode of responses that animals can show when they adapt to temperature gradients. And interestingly, as shown here by the black line that we evaluated with a meta-regression framework, there is a significant correlation between the strength with which they change their bodies and the strength with which they change their bills. That suggests that they can either be conforming hard to Beckman's rule, conforming hard to Allen's rule, or doing a little bit of both, which puts them in the middle, right? But there's really this restricted set of morphological responses is indicated by this relatively tight correlation that we see. So we think this is kind of interesting. But it really doesn't tell us why some animals do one or the other, right? And so then that brings us to my third point, which is what are the drivers of conformity to Beckman's and Allen's rules? So first, we can just extract the family level conformity and map it out across a cladogram here, as shown here on the top. And we see that most of the Beckman conforming families, as depicted in red, lie to the left of this black asterisk. That is, most of them are non-passerine lineages, right? These are the older birds, stuff like, what is this, pheasants, rails, hummingbirds, hawks, eagles, owls, falcons, woodpeckers. And most of the Allen conforming families tend to be to the right of the black asterisk of that node, right? We're suggesting that they are part of the passerine lineages, right? These are songbirds. But nevertheless, we do see that most families conform to neither, right? They're shown here in gray. And so we kind of thought about that and we're wondering why. So families that conform to neither rule seem to have fewer than expected species, whereas families that conform to either Beckman's or Allen's rules had more than expected species. And this kind of makes sense from a statistical sampling standpoint, right? Because if we only have a few species per family, we might not be able to detect a true conformity So yeah, sure, that makes sense. They might well be conforming to any of these rules, but if we only retain few species per family, we might not be able to detect that conformity. For kind of the same reason, families that only have a restricted temperature range, families that will conform to neither rule have narrower than expected temperature ranges, and families that conform to Bachmann's rule had broader than expected temperature ranges. Again, this makes sense because in order for us to detect a true conformity, we might want to sample across a wide space of this temperature gradient, right? Again, if our samples only conform a restricted space, we might not be able to detect a true conformity. What I think is then really interesting is that we looked at what the bill shapes of the different families that conform to these different rules. And we found here that families that conformed to Bachmann's rules tended to have rarer, that is more specialized, uncommon bill shapes. And you can see here when we plot bill shape across all species for which we have data, about 4,000, we can see here a strong segregation of the different species. So species that belong to families that conform to Bachmann's rule, shown in red here, they occupy the periphery of morphospace, right? They are not clustered around the center. They're out on the sides. They have weird bills, uncommon ones, right? Whereas species that belong to families that conform to Allen's rules, in blue here, they tend to be pretty clustered around your average bill size, right? They're kind of common bird-like bills. And for this result of rarity, so we could then quantify the kernel density within each of these morphospace plots, and we can show that there is a significant effect for the families that conform to Bachmann's rule. They have much rarer than expected bill shapes. And this makes sense to us from, when we kind of think about how birds use their bills, right? Hummingbirds and hawks and eagles all have pretty extreme bills, and they use their bills for their foraging lifestyles, right? Hummingbirds drink nectar, hawks and eagles use their bills to kill. We might think that they might not be able to retain their characteristic essential foraging strategy if they have to retool and change their bill size or shape. Instead, they perhaps might be free to change their body size as they adapt to a thermal gradient. And likewise, this is perhaps not illustrated, but we found that families that conform to Allen's rule have smaller than expected body sizes. Right? And so this is, again, we also interpret it in the light of constraints. So if a family has already a very small body size, perhaps they might be close to an absolute lower limit. So again, if they're tasked with the challenge of adapting to a thermal gradient, especially one to kind of hotter environments, if they might have the quote unquote opportunity, right, to change their body size, but if they're already at a low limit, they might not be able to shrink their body size anymore. Instead, they might be free to change their appendage length and conform to Allen's rule. So that's, yeah, and we do see that. So overall, I hope I've shown today that adaptation to thermal gradients can occur via alternative pathways, right? We don't have to always just be testing conformity to Bachmann's or Allen's rule in isolation. But mostly, we see that adaptation to thermal gradients is subtle and complementary, right? The adaptation is multidimensional. And last, I've shown that the process of adaptation to thermal gradients can be constrained in predictable ways by both phylogeny and ecology, right? So when we know something about what a bird family of bird is are like, we can predict what mode of response they're going to show, right? So with that, I want to thank a lot of people involved, especially Joan Garcia Porta and the rest of the Texas Botero Lab. Also Jonathan Myers, definitely, and the EPB program and DPBS for allowing me to continue my PhD here. And last, I want to thank Jonathan Moses for helpful comments on an early draft. And I want to thank you guys for your attention.
Great talk, Justin. I seem to notice in one of your plots that you use absolute bill size in your analyses as opposed to relative bill size, that is bill size relative to body size. I'm curious if you could elaborate on your justification for using absolute instead of relative bill size and how you expect the result to change if you were to use relative.
Great question. I unfortunately had to cut this out because of the expected time limits, but we did address relative bill size here. So yes. Yes. there's a really strong allometry between body size and bill size of birds, right? Big birds have big bills, likewise. So it makes sense for us to want to account for this allometry in a framework, right? And so what we did here is then just reran the same analysis for our that measured conformity to Allen's rule with the same approach, right? But here instead we fit not just bill size as a function of temperature, but we fit it also as a function of body size and temperature. But again, we allow for these phylogenetically pooled family level random slopes of temperature of bill size. Right? So then we extracted those slopes and we see largely the same kind of pattern, right? Most families are not conforming to either rule at the same time. However, we see more families, about 16 families end up conforming to Allen's rule, shown in blue here, 4 families that conform to Beckman's rule, shown in red, and also 4 families that apparently conform to both rules at the same time. But this might be this could change our interpretation, but we have 2 reasons why we don't want to do this, right? Because for the 4 families that show this spurious conformance to Allen's rule, right? These are owls, rails, hawks, pheasants. So they all increase their relative bill size with temperature here, shown by significant, varying degrees, but significant changes, their increases in relative bill size. All those changes are being driven by their conformity to Beckman's rule. So they're not changing their bill size, they're shrinking their body size while keeping their bill size changed, keeping their bill size the same. So, and the kind of 2 final reasons why we chose to not focus on relative bill size is that relative bill size is physiologically misleading. So 2 animals here, very different animals, right? Toku toucan and minute hermit. They have the same absolute values of their relative bill size, right? Which is, it's just residuals of the bill size versus the body size. So they have the same values, but they have wildly different thermal behaviors, right? Toku toucans can lose up to 400% of their body heat just via their bill, right? Hummingbirds, they barely lose any body heat through their bill. The few studies that have examined hummingbird bill heat loss suggest that heat loss in hummingbirds primarily goes out of a little bit of their face and from their armpits. A little bit comes out of their tarsi as well. But these are clearly very different animals, right? That are losing heat through very different parts of their body size. So we think that relative bill size can fail to capture that. Instead, we want to use absolute bill size. And then finally, this is just more of a labor point. I'm gonna stop there. It's a great question. Is that so if we imagine that if we can model, if we want to imagine that birds are just like globes, right? With kind of conical bill size, we can model, right, the effect of, or how well relative bill size and the actual surface area of the bill are correlated. And when you use absolute bill size, they're highly correlated, shown here in the top plots. But when we use relative bill size is poorly correlated. In fact, not at all correlated with the bill surface area, right? And the bill surface area is where the rubber meets the road. That determines the size of the thermal window, right? Relative bill size does not. So, thanks.
Great, thank you.
So, any more questions?
We have a lot of applause.
So, any more questions? Okay, I have a question out of curiosity. So, how is the aperture? So, penguin in your group, are they big like Allen's rule or the other rule?
That's a really good question. We decided to not include both of those groups because they're marine, and there seems to be a little bit more, let's say, diversity of opinions about how well the global climate databases can really capture marine climate. So we just decided to sidestep them and not include them. I could only speculate.
Okay, thank you. It's like the pigeon is a classic example of the body size change across latitude.
Okay. That makes sense. That I would suspect that they would, right? Because I mean they're pretty globular as it is, right?
Yeah,
So it doesn't surprise me that they conform to backgrounds real strong.
Let's say again for adjusting.
There we go. Thanks.
Okay.
great. Just do it from here. All right, cool. And how's this work? Yeah, sorry. Thank you. Shoot, sorry. Where is yours? Hi, y'all. Good to go? Okay, cool. Hi, y'all. My name is Sean McHugh. I am a second-year PhD student in Michael Landis's lab. And today I'd like to talk to you a little bit about biome shifts and how to model both realized and fundamental biome affinities, i.e. Shifts in where species actually occur as far as biomes and where they are capable of occurring. So briefly, I just want to kind of go through the spiel of like, what is a biome? And so here we have this famous plot from Whitaker of mapping a broad category of biomes over environmental space, particularly average precipitation and temperature. And over this environmental space, we see these ecoregions or large scale areas with distinctly different vegetation patterns from, as we can clearly see, there's a big difference between a tropical rainforest and a desert. Clearly, oftentimes, the species in these biomes have pre-adapted to or adapted to these environments. There is and shifts between these different biomes are not insubstantial. That said, we see species thriving well outside of their native biomes frequently. From the monk parakeet here, with being a fun example, including an anthropogenic biome, if you consider urban biomes such, with the monk parakeet going from Southern American temperate grasslands to urban environments. In this case, I think like Staten Island. Yeah. But there's many other instances of this too, where species are able to, once having access to a new biome, able to thrive within it, whether being shifting from temperate Mediterranean to desert, or temperate forest and tundra to grasslands, or tropical savanna to, well, everywhere in case of fire ants. But this really gets into the question of really not just what causes a biome shift over macroevolutionary timescales, a shift in where a species can have a sustainable, thriving population. You really have to wonder, what causes a species to not live within a biome, and what restricts a species distribution to specific ecoregions? And in this case, I'm going to take a figure from the species distribution modeling literature, BAM. And BAM is just breaking down the different components needed for a species to actually occur someplace. You have B, the biotic component, where it is shifting for where has to be able to compete appropriately. It can't be out predated. Biotic interactions need to be amenable. You also have abiotic component. It can't freeze, it can't cook, it can't desiccate. It needs to be able to tolerate and survive the abiotic conditions. And then also movement has to actually be able to get there. And many things can limit movement, large dispersal barriers such as mountain ranges, or even just sometimes, well, an ocean's a good one too, roads, depending on the scale. But with this, we want to break it down into two major components that are a big focus usually. The fundamental niche, where a species is able to physiologically occur, where are the abiotic conditions appropriate? Where can it survive? But then usually this wider distribution is truncated by a lot of biotic interactions as well as dispersal limitations to a realized niche. And that is often what we observe and what we actually collect data for. And in our macroevolution models, when we're trying to see where different biome shifts occur, we are only really focused on the realized niche. But however, species can disperse a couple different ways under this framework of fundamental and realized niche, right? So in this rough example of an environmental space grid, a temperate and tropical biome, and we have a species with its fundamental niche clearly in the temperate. In this instance, to shift to the tropical, you need a species that the species must adapt physiologically to the new environment, to the tropical environment before it can have access to it over millions of years, what's observable over millions of years. But also you could also have instances where it has a fundamental niche already in both biomes, and it's just simply a shift to extending, gaining access due to shifts in biotic interactions, shifts in dispersal limitations, maybe a long distance, very improbable dispersal event happened, finally allowing it to have access to this new biome. But again, as I mentioned, we only really look at the evolution of realized biome affinities. So I'm going to do a few mock simulations to show you what I mean with this. So with a common model that we use to model biome affinities, we are called the DEC model, accounting for dispersal, extinction, cladogenesis. Species can gain or lose biomes. So starting with an ancestor that's tropical, we then go and see a split over this small phylogeny where we have two daughter lineages that are both tropical. Then we see two gains where one gains a temperate biome, the other gains an arid biome. And then we see a splitting event in which cladogenetically the biome range breaks apart. One species occurs only in tropical and the other species occurs in temperate and tropical. This is a common thing we model. We're interested in these range splits. And then also you can have loss again. So it loses the tropical biome in both instances. And thus you have three species that occur in three totally different biomes. This is just a rough demo to show. But in this case, we're only looking at where we saw them occur and tracing that back in time, not considering where they could possibly occur. Where are they capable of occurring physiologically? So in our framework that we're proposing a new model that expands upon this framework to account for both the changes in the realized and fundamental niche. For this temperate species to gain a new biome, it must first gain a fundamental affinity. So in this case, we're representing that with an open circle of the same color. Then it gains a realized affinity, and then to lose, it follows the same stepwise order transition. Now, these events can happen very rapidly between each other. Of course, we can see instances where a species can enter a biome it doesn't do super great in and slowly adapt. But we're talking about explicitly here the realized affinity being where it has a sustainable long-term population observable over a phylogeny over millions of years. I want to make that clear because as you get into shorter time scales, the details get iffy. But take our same simulation again. And I added a few points for fill in the blank here. What would you expect to be occurring here to have that simulation we just ran through fit this framework of fundamental and realized affinities? So what we have here is, again, a bunch of open circles, but a key thing I want to highlight here is with cladogenesis, we expect the realized affinities to change. Biogeographic, that's part of the thing, the biome is part of the range, those split. But however, just because you have a vicariance event where a species no longer has access to the other biome, it takes much longer than an instant, very rapid speciation event for those abiotic, for the traits for the underlying abiotic tolerances to be lost as well. So we consider only range breaks in the realized affinities. And this gets into some interesting details here. This is just one example, but given, again, the same tip states, we have which of these scenarios is more likely? And we account for this using probability, of course, but I want to flesh out what these two scenarios look like. So one we could have is a tropical specialist ancestor, where it had a realized niche in a tropical biome, and it then split out and diverged to occur in multiple different biomes. Another potential scenario with the same tip states is having a tropical generalist, one that has a realized affinity. It occurs in sustainable populations in a tropical biome, but it has it is able to physiologically sustain itself in arid and temperate biomes as well. And and over time, it specializes into one of those, the arid, tropical, or temperate biomes. So these are two totally different scenarios with pretty divergent evolutionary implications. So being able to account for which of these scenarios occur is quite valuable, or at least characterizing the uncertainty. Of course, one big thing here is those fundamental affinities are probably going to be hard to come by, but we have a lot of instances that we just simply don't plug into our phylogenetic models, from invasive species, some physiological experiments that have been done, in ways we could, and also fossils to some degree, ways to account for these fundamental affinities would be quite valuable. Especially over larger trees where you could better characterize this. That said, how are we doing so far with developing the model? So this is we're on the early stages of this, but so far we can, with a single rate parameter describing this process, we can accurately recover the parameters that we simulated data sets on. So what we're doing here is we're simulating trees just like we showed before. That's a process of generating gains and losses of realized and fundamental biome affinities over phylogeny, and then fitting our model in a likelihood-based framework to see if we can recover the simulating, the generating parameters as the when estimating against those tip states. And we did pretty good. But our interest now is more so in understanding what is the differences in rates between different categories. Do we gain and lose fundamental affinities at a faster or slower rate than realized affinities? Does the physiological conditions change more than the spatial biome patterns? Or do gains and losses different patterns? Or do different biomes have just different context-dependent scenarios where this occurs? So getting into those details is what we're really excited about, and tying this potentially even to diversification as well. So in general, though, we also have a set of future directions we're pretty interested in. One is we really would like to fit this to an empirical example. In this case, by Burnham, I gave this very open source Google image you just shown, kind of the array of them, but this is a relatively diverse clade, I think about 150 species, and they have realized affinities for a wide range of biomes, and with some having affinities for multiple biomes and some having affinities for just one. Seeing what the fundamental affinities amongst these in a clade with a lot of variation and diversity and realized affinities would be quite interesting. Additionally, we are interested in hypothesis testing. Again, as we mentioned before, one of the sets of rate categories we're interested in, which biomes are easier to gain or lose, both in fundamental and realized affinities. And then additionally, I'd be really interested in modeling hidden fundamental affinities. If we consider, say, different urban environments, say like urban biomes or even indoor biome, I've seen some literature on that as well. It'd be pretty interesting to predict which species, given the ones that already occupy urban biomes, which other ones, using phylogeny, which other ones could have that fundamental affinity and be prime candidates for dispersing to these new biomes, to these anthropogenic biomes. All questions we're very excited in, but and happy to take questions as well on this. I prepped this as a shorter talk, so I'm going to roll into acknowledgements now. Thank you so much to all members of the Landis Lab for their immense support and patience. Thank you so much to the Living Earth Collaborative for the opportunity to present. Thank you to Worcester, DBBS. I don't have that emblem on there. I apologize. But anyways, questions?
Hey, John, really nice talk.
Thank you.
Super exciting work. So I see that you are sort of leaning towards this direction of incorporating hidden states, which is super cool. And I was kind of curious, are there ways that you're thinking about incorporating extra phylogenetic sources of evidence for these different sexual biome states, things like the retention of particular ancestral states
Mhm.
in the among sand species. So on top of that, are you sort of injecting paleo biome data into your model as you go along at ancestral nodes in the tree?
Yeah, that's super cool. Just yeah, that's exactly what I meant when I mentioned fossils before. There are some fossil taxa we have paleobiome data for that can help better leverage which scenarios there are certain realized affinities to help constrain and better trace back. By other things, I assume you meant other traits in concert with biomes like topaz or other traits like that. I definitely agree. That'd be super interesting. And also what you mentioned with hidden states, I could definitely see, just in addition to fundamental being an hard to observe state, not necessarily directly unobserved. I would say adding in actual hidden states for accounting for other forms of rate heterogeneity across these would be very interesting, because there's a lot of, as with most discrete trait evolution, there's a lot of context dependent stuff that can change rates that is unobservable at all. So that would be a very cool thing to pursue as well. Great questions. Thank you. Hi.
Hello, I have a very basic question.
Yeah.
I was wondering, how do you know when a species has a fundamental state or a fundamental affinity for a biome when they do not have a realized affinity?
That's a great question, and that can be a tricky one to get data for, of course. A big one is seeing from invasives, was it able to invade, rapidify, and thrive within that biome? Experimental work is another approach, seeing like within those abiotic conditions, how well is it able to survive? How much does different key performance decrease? That's a tough one in general. A lot of the fundamental niche stuff often emerges from like continuous traits like temperature or such, and those are equally hard as well, and usually same approach, seeing if they've been able to have, when access to those different areas, are they able to survive or not, or are they able to thrive? And same with experimental approaches. Great question. Thank you.
Great talk, thank you.
Oh, thank you.
I have a question about whether you're including different resistance of transition from one biome to, like, say, A biome to B biome.
Yep.
I'm thinking about like more temperature of species that have a higher optimal temperature, usually have narrower range.
Mhm.
than before, and I'm just wondering what you fit in your model.
That's a great point. So one way we could account for this broadly with different rates is saying you have different rates of transitionings from one biome to another. Like it's a lot harder to transition from a desert biome to a tropical biome, so on and so forth. You could tie this other with key traits or other metrics to distance, like, well, if we were to refer back to the Whitaker biome plot where they map them, what is the climatic distance between those biomes, and incorporating that in as well. We've done that a lot with these sort of discrete state models with biogeography, where having transitions be distance dependent. You could do similar frameworks here. That's a great question.
Nice talk.
Hey, thanks.
A couple questions. First, I'm curious, how much additional information do you need besides the current occupied niche for each species tip, I guess, on the tree? Maybe you can just talk about it in your model. And then so far, have you modeled like trees with 3 tips or larger?
Much larger. The idea is using it on ideally phylogeny with like hundreds of tips. That'd be the dream. And in those cases, we might have some that we have some sense of the fundamental affinities, and others we have none. And we'd like to be able to bolster the analysis with which tips we do have that data for. We'd ideally like to have it for some, but even if not, just the structure of the realized affinities, given that you must first transition to a fundamental affinity before a realized affinity, that constrains at what sort of fundamental affinities are possible to some extent, especially over larger phylogenies. Does that cover the question?
So because of the time, that's same showing again, and we can.
Awesome. Hi, everyone. My name is Rhiannon Vargas. It is really great to be here in person. I'm a 5th-year EPB student. And so this Thursday seminar is, is quite a tradition. And, I really love what Wen had put together, the idea of graduate students coming to talk about their diverse research. And I think that, I'm going to give you a little taste into the micro world now with ecology specifically. And yeah, I'll just go ahead and get started. So today I'm going to be telling you about a very specific project for my thesis that revolves around dairy cows and dairy farms. But I also wanted to just briefly introduce my research interests. So overall, I would say, generally speaking, very interested in microbial ecology, and I'm particularly interested in the ability to predict these microbial communities, how they assemble, their functions, and being able to do that with tools and applying them to diverse systems. And so pictured here are just a few of the different projects I wanted to show, but I think that's one of the most interesting things to me about this, and you can apply these analysis and tools to many different things and very similar questions. Like I said, today I'm going to be telling you more about cows. So why dairy farms? The dairy farm environment is really interesting to study for many reasons. And one of them is the functional aspect of the bacteria that can be found there. It's really well known that antibiotic resistance is a really big deal. And it's also well known that farms are characteristics of potential transmission events. And so this is a scary number to hopefully get you to understand a little bit more about why this is an interesting ecological area to study, in that it not only is contained within the farm, it could there's no geographic boundary, right, with this resistance. So 2.8 million antibiotic resistant infections occur in the US each year, and more than 35,000 people die as a result. It's also very costly. And like I was mentioning before, dairy farms and just agriculture in general, it's just well known that they are using these antibiotics. And in the US, there are no regulations or policies that farmers have to report how many antibiotics they're using. So like I mentioned, you can imagine that there's this transmission going on from farmer to potentially, our community that is really based around the dairy farm, and there's no geographic boundary for this. So it could really escalate. Okay, so people have looked at this before, and what you're looking at right now, and I'll walk you through it, is a project that I was on with Kim Sukum. She was a postdoc and instructor in the lab, also was a former UPB grad student. And so we were looking specifically at how manure and its taxa and functional profiles could differ between both fertilized soil and also basically how it was being stored. And so even though people have studied similar systems like this, nobody has looked at it at this depth. And every time people go ahead and research this, they find new things, new resistant genes. And also a lot of the databases out there to characterize these resistant profiles and taxa are very human dominated. So there's still a lot to be put together. There's a gap in this information. But what I wanted to show here is that we found that there is this reconfiguration of both taxa and functional profiles when you take this fresh manure and store it. So there are potentially a lot of different sources that could be taken on this farm, right? And so here are some figures showing the species richness and beta diversity differences of fresh manure in red, and then the manure pit at different depths, and is significantly different from soil. I feel like my earring is okay. Anyway, And we also found the same result when we looked at the resistant genes. And so this was interesting because it let us know more about what is happening on dairy farms and different parts of the farms. But additionally, what was lacking in the study was the farm worker component. We knew it was happening in the manure with the potentially the fertilized soil, but we weren't really sure about that transmission aspect. And so that leads to the hypothesis of this project that I'm going to be telling you about. So we predicted or hypothesized that there is a microbiome overlap between dairy workers and dairy cows due to occupational exposure, which would differ significantly from control office workers serving as the control group. And the second hypothesis being that dairy farm workers would harbor significantly similar resistomes or antibiotic resistant gene profiles to those present in dairy cows that they manage in their operation compared to office controls. Okay, so we call this DOME for just dairy-omics, but I might refer to it as DOME. And this is the overall experimental design. It was a longitudinal study that was expected to take over the course of 3 years. We have seasonal information, and we recruited dairy farm workers and then the dairy cows on their operation, as well as sex-matched and age-matched office workers. And we collected both fecal samples from previous, the reason being that previous studies have shown that the fecal, the nasal sample microbiome might be more susceptible to these particular environments, as well as the fecal sample. So we had 2 different types of microbiomes for an individual, and we had competitive sampling. And what did we do with that type of data and those samples? So we were interested in, one, characterizing the bacterial community. So we did that with 16S. We were interested in looking at function, specifically antibiotic resistant gene profiles, which we did with metagenomic sequencing. And then the functional screening of antibiotic resistant genes was to characterize novel resistant genes that we do find. And then the cultureomics aspect was, I'm not going to talk about that today, but specifically looking at particular bacteria and how they might be transmitted. I got to say that this is a big collaborative effort. I didn't collect these samples. Our collaborators in Wisconsin did, and I couldn't have done it without the help of these fabulous people, helping processing hundreds of samples, and then doing different parts of the pipeline. Okay, so what I am going to be talking to you about is the 16S taxonomic bacterial profiling I did of these 2 different sample types. And then I'll briefly go into some of the functional resistant gene stuff that's still a little preliminary. So the main part of my talk is going to be the taxonomic characterization of both the fecal and nasal samples across hosts. And so we had over 600 fecal samples and 600 nasal samples to compare. And I ran it through a software called Data2, which was able to give me amplicon sequence variants to compare. Okay, I'm just going to throw you into this data. I'll walk you through it though. So what you're looking at is a stacked bar plot of taxa. And really, you're just going to see big patterns here, right? So on the right, you have the farmers, and on the left are the office workers. And this is specifically from their nasal cavities. These are the top 30 taxa. Those in yellow are considered all the others, obviously more than 30. And the main point I want you to take away from this is that dairy farm workers have a lot more color going on. They also have a lot more others. So their percentages of these top taxa are different than office workers. And particularly what I want to put your attention to is that blue line on top of the farm workers stack bar plot that you don't see in the office workers. And what that blue line is a Ruminococcaceae taxa, which is found in cow manure. Similarly, now you're looking at the nasal stacked hormones for cows. I'm showing you this because very similarly, they have Rhiannon, from manure just kind of going in their noses, but it's also very diverse, much like we're seeing with the farm workers. And so when we look now at the alpha diversity differences and the significant differences between those, we do see that there is a significant difference between cow nasal diversity and office workers there in orange and blue respectively. There's a significant difference between the human groups, or between the blue and the farmers in green. And there's not a significant difference in diversity between the cows and the farmers. Okay, so what does this mean? I know there's a lot on this slide, but I was able to do a general linear mix model to look at specifically what are those differences, what are those significant differences. And what this plot is showing is relative to control office workers, where do those farmers taxa fall? And so there's a lot to read there, but I just wanted to show you that there are a number of taxa, which if it's a positive value, means that it's significantly more abundant in the farm workers. And here in this case, office workers have significantly more Corynebacterium, which if anything just is a common nasal microbe that hangs out. And I picked out a few specific taxa here. Again, that rumen caucasiae. Again, it should be surprising because this is found in the rumen of cows, but also being found in the nose of these farm workers, significantly more abundant in farm workers than office workers. And then the true is for it's also true for other taxa like Clostridium, which is known to cause different diseases. So I put the fecal stuff last just because it was semi-unsurprising that the humans and cows are very different. And so this is the same alpha diversity plot, but now with the fecal stuff on the left side, you can see that cows just have a lot more going on in their gut than humans do, and that the humans are pretty similar. Again, not going to spend too much time on this, but I wanted to emphasize that we're not seeing really major big picture patterns in the fecal microbiome between the two human groups. And they're dominated by common members of the mammalian gut. The cows are also pretty similar across the board. This is pretty interesting. There are repetitive samples in here, but this is from different farms. Very surprising that across the board, they're pretty much dominated and look homogeneous. Okay. And I would say that this is probably my takeaway slide right here to summarize everything that I've told you so far. So what we're looking at is beta diversity differences between all of the samples for bacterial 16S sequencing. And the main two things I want you to look at are the color and the shapes. So in circle represents, the circles represent all of the fecal samples, and the triangles represent all of the nasal samples. And the colors are the same from the previous plots: cows in orange, office workers blue, and farmers in green. And I want to draw your attention down here to the fecal samples. Like I said, humans are pretty similar with humans, and the cows are pretty distinct on their own. However, when we go and look at the nasal microbiome, there's a lot more overlap going on. And specifically with the green and the orange, a lot more than the orange and the blue, which are the office workers and the cows. And even when we run this analysis with the nasal samples only, we still see the same patterns there, where we're having that overlap with the farm workers and the cows. Okay, so I talked to you a bit about my dive into bacterial taxonomic classification, diversity analysis. I'm going to tell you a little bit about what my coworker, co-lab member did for analyzing the resistome. So first of all, we were not able to characterize the resistant gene profiles of our nasal samples because there was a lot of human contamination. And so we were only able to do this deep type of sequencing on our fecal samples. And so what Bejan did is he pretty much did very similar things to what I had shown you with diversity and with comparing between and within samples, but now he did it with specifically antibiotic resistant gene counts. And overall, we found that human resistome, so the resistant genes characterized in their guts, are richer than cows. And pretty much it's true for across season. And while it's very subtle, the farmer gut resistome is different than that of office workers, but it's still telling a very similar story with our fecal taxa, where it seems like it's more of like a humans versus cows thing. And just like a little more insight into what this is comprised of, you'd see that the antibiotic resistant genes that they're acquiring are pretty diverse across the board. But like I said, even though cows may be enriched in a particular antibiotic, it's still the human resistome that's more rich. Okay, so to summarize everything, there are significantly more like different taxa in the nasal compartments of farm workers than there are in the office workers, specifically with Ruminococcaceae, which is found in the manure, and also found in the nasal samples of the cows. So we know that fecal samples cluster by hosts, and this is true for the antibiotic resistant genes as well as for taxa. And our findings suggest that the nasal microbiomes are more susceptible to the occupational environment than the fecal samples are. And so we have some future directions to incorporate more metadata into this, such as looking within farms, but overall, I think it's additionally, we're only going to be looking at this year of sampling and hopefully publishing on it. So thank you all for listening. Thank you to the lab. Happy to take any questions.
That was really interesting. I think it was the figure you have about the resistance from the cows across seasons. I think it said that summer was not different. Do you have any rationales like why or thoughts?
I don't know why, but farmers have specific seasons that they go and work in. So they're 365 days of the year. They'll go and come. So I haven't quite investigated this, but we're thinking it might have to do with behavioral patterns that farm workers and humans have in general, specifically within the summertime, but specifically, I don't know.
Hi, Jasmine from YouTube is asking, do you think these results indicate an occupational hazard among dairy workers? Are these nasal bacteria that overlap with cow microbiome bacteria harmful to humans?
So the question is whether the bacteria that are found in the nasal cavities of farmers are potentially hazardous. That is a good question. So one thing about this analysis is we are looking specifically at, any DNA that is there. We don't necessarily know if it's viable, but what the interesting thing is that potentially genes could be shared even if the DNA is not viable. So a lot of this stuff is potentially harmful to humans. Whether or not they stay alive in an aerobic condition is unknown. But from some of the things that I looked at, they are potential pathogens that are opportunistic.
Okay, I think do you know if you transfer a patient from office to the farm, how long can the microbiome community stay there? or it will just change in weeks or months?
I don't know how long it would take to change, but there was this interesting study that looked at pig farmers. And they took sampling before their work shift and after their work shift, and then also after their weekend. And even within that short amount of time, it seemed like there was a lot of variation in what was being found in their nasal cavities. So I don't know for sure, but I do expect that since it's just an open area, that a lot of the stuff happening and is found there is constantly shifting. So whether there's such thing as a stable nose microbiome, I'm not sure, but it's a great question.
Thank you. And let's say Rhiannon again. And so please join. Next week, we also have 12 students who give a presentation. Please join us next week. Thank you.